and beep. There we go. Okay, so we've worked out that there's this stuff called DNA. It's the transforming principle. It's got a shape to it. In particular, it's a polymer, or to be more precise, a double polymer made up of a bunch of monomers called nucleotides strung together. And the shape that the entire thing takes is called a double helix. Am I doing this right? Hang on. Sorry, I just want to make sure I'm presenting this right. And the shape that the whole thing takes is called a double helix. A colleague of mine used to have a cartoon on her door that showed Watson and Crick walking into a bar, looking and despondent, and all of a sudden the guy next to them in the bar says to the bartender, "I'll have a double, Felix." And Watson and Crick suddenly look up. Ooh, double, Felix. That gives me an idea. Anyway, the reason all of that seeming trivia makes a difference is that to understand how everything shape of things. And each nucleotide consists of a phosphate group, kind of consisting of a phosphorus atom and some oxygens. You know, that phosphorus in schnapps, if you remember. In light green, we've got a sugar group. In this case, a sugar called deoxyribose. That's the light green. And then each nucleotide can have one of four possible bases, first, second, third, or home plate. Actually, I'm kidding. Adenine, guanine, thymine, or cytosine. And the bases are either single rings of atoms or double rings of atoms, mostly carbon and nitrogen, with maybe a few hydrogens and maybe an oxygen thrown in for flavor. So that's what DNA is. It's a double polymer of these nucleotides. What locks those strands of a DNA molecule together are some easily broken chemical bonds called hydrogen bonds. We don't really need to worry about how those work. What's important for our purposes is that hydrogen bonds are very weak, they're easy to break, and they're easy to reform. You could think of hydrogen bonds as being a little bit like Velcro, right? Sewing together two pieces of cloth takes a lot of energy, and it takes a lot of energy to rip the seam. But if you have Velcro, you can take two pieces of Velcro will stick together. It's very easy. You could think of it as being like two halves of a zipper. Of course, you know, cutting across a zipper is very difficult to do. You'd need pretty heavy shears to do it. But the point of a zipper is that the two halves are very simple to zip and unzip and rezip. And DNA is kind of like that. The sides of our DNA ladder are put together pretty firmly, but the hydrogen bonds that lock the bases together are easy to break and easy to reform. Because of the way the bases are shaped, an adenine on one strand, an A, can only form hydrogen bonds with a T on the other strand. Adenine can only form hydrogen bonds with thymine, and guanine can only form hydrogen bonds with cytosine. So an A on one strand can only form a bond with T on the opposite strand, and T can only, and then a G can only form with C on the other strand, and C can only form bonds with G. And that's ultimately because of the shape of those bases. And we call the whole thing base pair complementarity. This is why Chargaff's rules work. The percentage equals the percentage of T is that every T on one strand must have an A on the other strand, and every A must have a T. If it's not there, the double helix won't form properly. It won't have the right shape. You'll end up with something like, you ever have a zipper with one tooth that's out of place? Really hard to zip and unzip. Something kind of similar happens in DNA strands where the base pairs don't match each other perfectly. And the same is true for G and C. All right, if you just take that little strand right there and count, you can see that we've got 16 bases. We have 6 G's and 6 C's and 2 T's and 2 A's, right? So the percentage of T is what? 2 out of 16. That's 12.5%. And so is the percentage of A. And the percentage of G is 6 out of 16, which is 37.5%, I think. And so is the percentage of C. That's why Chargaff's rules work. It's because DNA molecules have to show that base pair complementarity. And it's also the key to DNA replication, because what goes on in that mysterious S phase of interphase that we talked about Wednesday is DNA replication. Every chromosome is basically one great big long piece of DNA. Each individual chromosome might consist of several inches of DNA. If you could somehow unravel it and lay it out all together. And what happens in that mysterious S phase of interphase is that DNA replicates. And when that happens, when DNA is duplicated, those hydrogen bonds that hold the two strands together are broken. We'll talk a little bit later as to what it is that's doing the breaking. The two strands that are unzipped move apart from each other. And now each one of these strands can serve as a pattern to make a new strand that matches the other. So new strands are assembled and each one uses the old strand as a pattern. So if we add nucleotides and fit them together to match the strand on the left, they'll look like that. And if we add nucleotides on the right to match the strand on the right, they'll look like that. And so our end result is we have two identical pieces of DNA because each of those strands can be used as a template to make a complementary strand. So you can take a piece of DNA and you can copy it, and you can copy it very close to exactly. You can make errors in DNA copying. It does happen, but you don't make an awful lot of them. Most of the time when you copy a strand of DNA, you'll end up with two strands that are identical to what you started with. And it looks a little bit like this. Here you've got a double helix. You can see up in about the middle of the page, it has unzipped. And then each strand can become the pattern for assembling a new strand. What's doing the assembling is a specialized protein molecule called DNA polymerase. There's actually several DNA polymerases that are doing that. That's a type of protein called an enzyme, and we'll talk Monday as to what enzymes are and what they do. For the moment, you can think of these as little fairies that do the work. That's not really what they are, but we'll get into what they're really doing on Monday. So when DNA replicates, it unzips, the hydrogen bonds break, and then each strand becomes the pattern for assembling a new strand. The reason that matters is that just like the order of letters in a word determines what word you've got and what the meaning of your utterance is. The order of nucleotides determines what information the DNA is capable of carrying. It comes down to the order of those nucleotides. And so we now know that every chromosome is a single long piece of DNA. When a cell is in interphase, the DNA is unwound, and that means you can't see individual chromosomes. When you're looking at a nucleus in interphase, you can't see individual chromosomes for the same reason that when you're looking at a big plate of spaghetti from a distance, you can't count the individual noodles, right? What you see is basically one mass of pasta-ness, but you can't really count the noodles. You may not even be able to make out clearly that there are separate spaghetti noodles on your plate. But at cell division, the DNA coils in on itself and wraps itself around a protein core, wraps itself around some specialized proteins that happen to be called histones. And this is an actual chromosome imaged with an instrument called a transmission electron microscope. And this chromosome has been partly unwound. What you'll get is the DNA will wrap around histone proteins and make what looks kind of like a chain of beads. And then that chain will coil on itself. And then the coils will coil on themselves. And then the coils of coils will coil in on themselves, making an extremely compact structure. That's the chromosomes when you can see them. They're always there, but in interphase, they're not supercoiled, and so you can't make them out as individual things. At prophase of mitosis, the strands of DNA coil around these proteins, and then the coils coil, and then the coils of coils coil, and then the coiled coils of coils coil, and make this very compact thing. And this is a chromosome that has been partially uncoiled. There's a chromosome where all of the DNA has been unwound, and this may be a little bit difficult to see, but down in the lower part of that picture, that kind of darker, cobwebby looking stuff is the remains of the protein, and then all of the gray stuff is strands of DNA that have been unwrapped around those specialized histone proteins. Up at the top of the picture, you can see some individual strands of DNA. All of that gray mass that takes up most of the picture, that is unwound DNA, just lying around in a heap like a giant plate of spaghetti. So that's what chromosomes really are. And DNA replication is what happens during that S phase of the cell cycle. Each chromosome replicates, it unzips, and it will actually do this at several points along its length. It'll untwist, unzip, and replicate to form two identical copies joined at the centromere. So every chromatid is identical to its sister chromatid. So the chromatid on the left here is a very long piece of DNA that's identical to the chromatid on the right. Okay? And so when this thing separates in anaphase of mitosis, each cell gets one identical copy of each chromosome. It's because the sister chromatids are identical to each other. And that's made possible by DNA replication. The reason that matters is that there is something critically important about the order that the nucleotides are in. You can actually go, if you really wanted to a website called NCBI, the National Center for Biotechnology Information. It's maintained by the National Institutes of Health, part of the US government. And it's www.ncbi.nlm.nih.gov. And stored there is a ridiculous amount of sequence information about DNA. We can take DNA and literally read the order of nucleotides of a DNA strand. Something called the Human Genome Project was finally completed in, I want to say, about 2005, when using a really neat technique called PCR that maybe we can talk about later, scientists were able to take the DNA of a human and read all of it. And this is a tiny piece of the data that's there that you could actually download and search through if you wanted from the National Center for Biotechnology Information. Somewhere in your DNA, there is a piece of it, and one strand begins CAGGGGGGGGTAAGGGCGGATTACCATTATTACCGTCGCGCAGGCT. I hope you memorize that. Actually, I'm kidding. Please don't memorize that. This is just by way of showing that there's something important in the order of nucleotides. Some early folks in the 1950s used to think that DNA would just have AGCT, AGCT repeated over and over again. Some people thought that the order might be random. It turns out it is neither. It is not random, and it's not the same thing repeated indefinitely. Information is carried by the order of nucleotides, analogous to the way that information is carried by the order of letters in a word. You can take, let me see. Yeah, you can take the phrase sweltering heat, rearrange the letters, and it will spell the winter gales. True. Try it yourself. Sweltering heat can be rearranged into the winter gales. Clearly, the difference between sweltering heat and the winter gale, not the letters, it's the order that you put the letters in. And depending on what order you put those in, you can make it mean completely different things. You know, just like, violence, VIOLENCE. Change the order of the letters and it spells nice love. So clearly, again, the order of the letters is what's carrying the meaning right here. Change the order and you change the meaning. And you might get something completely meaningless, but you might get something very different. Right. To understand what the order means, we have to look at a second major class of biological molecules, and that's the proteins. Nucleic acids are important in heredity. You can't have heredity without nucleic acids. Proteins are important for every bleeping thing that a cell does. Every living cell, even the simplest bacteria, has to make thousands of different proteins to run its metabolism and reproduce and carry out all of its life processes. So, for example, we talked about animals have an extracellular matrix made out of collagen. Collagen is a protein. Your tendons and your ligaments, like the Achilles tendon at the back of your heel or the nuchal ligament at the back of your neck, are made up of sheets of tough protein, made up of collagen. Keratin is what your hair and your fingernails are made of. That's a protein. Your muscles are made up of something like 50 proteins, but the ones that do most of the work of actually contracting are a pair of proteins called actin and myosin. We probably won't have time to look into how they work, but they're proteins, and they do all of your muscle contraction. The proteins that drive chemical reactions are called enzymes. That may be a word you've heard before. Those are proteins. The stuff in your stomach and intestine and even in your spit that breaks down the food you eat into small molecules that your body can absorb. Those are enzymes, and they're proteins. Signaling proteins like insulin. Insulin is released by your pancreas and basically says, hey, all of you cells, we've just had a meal. There's a lot of sugar coming your way in the bloodstream. Get ready to absorb it. If you can't make insulin or your cells don't respond to insulin, you are a diabetic and you have to get insulin injections. That's a protein. The hemoglobin that carries oxygen through your blood, that's a protein. And there are a ridiculous other number of proteins that do all kinds of things. Every cell has to make thousands of different proteins just to do its basic metabolism and just to grow and replicate and do whatever it is that it does. So what's a protein? Proteins are polymers of a different type of monomer. Come on, Wagoner, drink some coffee. Nucleic acids are polymers of nucleotides. Proteins are polymers of amino acids. And there are, for your purposes, 20 different amino acids that are used to make proteins. There are actually lots more amino acids out there, but only 20 of them are used to make up proteins. You may have heard, if you know anything about nutrition or you studied it, that there are some amino acids that are called essential amino acids. I believe that's 8 or 9 amino acids are essential, meaning that if you don't get them in your diet, you run into trouble because your body's not able to make them from anything else. The other 12 or 13 amino acids that are not essential, if you don't get them in your food, your body is capable of making them from something else. But you've got to have a supply of all 20 amino acids, whether you're getting them from your food or whether you're making them yourselves. You've got to have all of them in order to make up proteins. Okay, you make a protein by putting amino acids together in an order. The order ultimately determines the shape of the protein, and the shape of the protein is critical for determining what the protein does. That's the basic message of most of what we're going to be talking about for the rest of the day and also on Monday. As I've said, there are thousands of amino acids. Red Bull contains an amino acid called taurine. Taurine, I'm not sure how it works in keeping you awake or anything like that, but taurine is not necessary for making proteins. You don't make proteins out of taurine. There's some others that you'll come across in, there's one called carnitine that you'll see on other brands of energy drinks. Ornithine is another one that you might encounter in energy drinks and supplements and things like that. Those are not part of the magic 20. There's only 20 amino acids that are used to assemble proteins. There are others that might have other uses, but only 20 are used to assemble proteins. So just as a nucleotide has 3 parts to it, a sugar, a phosphate, and a base, an amino acid is defined by having 3 major parts. It will have a group of atoms consisting of a nitrogen and a couple of hydrogens. That's called an amino group. It will have a carbon with a couple of oxygens stuck to it called a carboxyl group. And then it will have an atom or a group of them called the R group. And it's different R groups that make different amino acids. The one on the left in this diagram That amino acid is the simplest one. Glycine, the R group is a single atom of hydrogen, shown in green. The one on the right, I think that one is the amino acid valine. I could be wrong about that, but it's a different amino acid. And the R group is a chain of a number of carbon and nitrogen atoms with some hydrogen stuck on. It's a very different R group. And different amino acids have different R groups. The R group is what makes amino acid different from each other, but what they've all got is amino and carboxyl groups. Just as all nucleotides have a sugar and a phosphate, and what makes them different is the base, what makes amino acids different from each other is they've got different R groups, and they all have a common amino group and a carboxyl group. That's what they share. Here's some of the 20 main amino acids, and by the Allfather, I don't want you to spend any time worrying about this. All I want you to see is that up at the top of each one of these diagrams, there's an amino and a carboxyl group. And then shaded in green in this case, we have different R groups. More with the R group shaded in, and there, and so if because you're going to need a lot of R groups. Thank you very much. Thank you, both of you. Okay. You certainly do not need to memorize these amino acids. They've all got names. There's arginine and histidine, aspartic acid, glutamic acid, and lysine, and asparagine, and cysteine, and glutamine, glycine, and serine, and threonine, and tyrosine. Just know that there's 20 of the bleeping things that we are concerned with when we're talking about protein synthesis. Amino acids can form a bond between the amino group of one amino acid and the carboxyl group of another. On the left in red, we have an amino acid called aspartic acid. Looks like this. On the right, we have an acid called phenylalanine in blue. Now, we've lined them up so that the carboxyl group, that carbon with a couple of oxygens on the aspartic acid, is right next to the amino group on the phenylalanine, and we can make a bond form between the carboxyl group of one amino acid and the amino of another. So now there's a new sticky bond between the carbon on a carboxyl and the nitrogen on an amino group, and the extra atoms fall off and they form a molecule of water, and we call this bond a peptide bond. A peptide bond is a bond between the carbon of a carboxyl group and the amino, the nitrogen on an amino group. And you'll notice that we have a carboxyl group over on the right that could bond with another amino group, and we could add a third amino acid if we wanted to. And then that amino acid would have a carboxyl that could stick to another amino, and then that one would have a carboxyl that could stick to another amino, and we could string these together for, I don't know what the real maximum is, but we could put thousands of amino acids together all in a row with the carboxyl of each one bonding to the amino of the next one. And so we can make chains of amino acids of indefinite length. Incidentally, there's a reason why I picked these two amino acids. If you just stopped there with an aspartic acid stuck to a phenylalanine and stuck just a few more atoms on one end, you would have a compound called aspartame, otherwise known as NutraSweet. The no-calorie artificial sweetener in Diet Coke and other diet beverages, for example. There's concern about the health risks of consuming aspartame. I'm not really up on that. It's probably bad for you to consume too much of it, so you probably shouldn't slam 3 2-liter bottles of Diet Coke a day. That might uninsulate your wiring. But in and of itself, aspartame is basically 2 amino acids stuck together with a peptide bond. So now you know. All right, we're keeping on going. When we talk about how a protein is shaped, we can talk about 4 different levels of structure. The order you stick the amino acids together in is called a protein's primary structure. So if you take a bunch of amino acids and you put them together, a methionine and then a cysteine and then a threonine and then a tyrosine and then a glutamic acid, dot, 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 the order in which those amino acids follow one after the other is the protein's primary structure. When you've put amino acids together in a chain like that, the effect is a little bit, you ever have presents and you've got a long piece of tape that just won't stop sticking to itself? Chains of amino acids act like that. The amino acid chain will start folding and bending and kinking, caused by electrically charged atoms being attracted to each other, almost like static cling. And so parts of an amino acid chain will fold on each other, and that secondary structure. Secondary structure tends to affect just local regions of a chain of amino acids. Then there's more folding caused by R groups being electrically attracted or repelled by each other. So you start with a linear chain of amino acids, but then with secondary and tertiary structure, that refers to parts of that chain being attracted to each other or repelled from each other and sticking to each other. So that causes your chain of amino acids to fold up into a very complicated shape. In fact, it's actually a major problem in computer science, is that we can't predict how a chain of amino acids amino acids together. This is a computer model of what a simple chain of amino acids would look like.
What happened?
here. Okay, one person's still here. We lost connection in the middle of that. If you're listening, I'll just pick this up next time.